All right, while waiting for the computer to turn on, I can make the announcements. So your third assignment is already posted on the website, although the second assignment is due tonight. For those who have finished the second assignment, they can start working on the third one. It's due on May 9. The 9th of May is a Monday, so it's after the deadline of second assignment. You are going to have 11 days to work on the assignment. I have written the solution for assignment 3 yesterday. My code was, I didn't think about creating efficient or clean code, so it contains lots of if else checks, and it just has a single function. At the end it became like a 500 lines of code, but I was able to finish it in 4 hours. However, in terms of lines of code and the effort, in terms of complexity of the code, it is going to be as complex as the second assignment, although Viterbi is really clean. What was difficult in my coding was that the fact that the hidden Markov model, there are certain states missing. Okay, for example, match state is followed by 3 states, the 3 previous match, insert, and delete state. But in the input HMM, it may be the case that some of these states may be missing. They may not be needed, and such cases made the programming difficult. Okay, that was the part that was difficult to implement the Viterbi algorithm, the forward algorithm, was the fact that some states was missing. But here's a hint for you, which you can even produce much more efficient, less lines of code. A better coding approach should be, after reading the hidden Markov model from the input file, you can just fill in, although there are missing states, you can just put those missing states in your hidden Markov model and just do not have any, their transition, in the transition table, just set their transition to 0. Okay, so in that case, you don't need to think about special cases. Every state is going to be preceded, every match state is going to have 3 preceding match, delete, insert states. They may not be existing, some of them may not be existing in the input hidden Markov model, but you are going to just put them there, and you are going to set the transition probabilities to 0. And which is not going to change any computation, and you will not need to consider any, you don't need to write many if statements saying that, okay, I had if statements like that. If all 3 previous states exist, do this. If these 2 of them exist, use this formula. If for example, if all match, insert, delete exists, the formula is this. If match and delete exists, use this formula. If these 2 exist, so my code was like this. It contained lots of if, else, and considering all. That was the reason it was 500 lines. But I think this hint, even you don't have some of these states, even if they do not exist in the HMM, you can just put them, but you can construct a matrix on your own, which is actually going to have more states than the given input to the Markov model, but just you are going to have 0 transitions to those states, and it will make the forward algorithm implementation much easier. It will be a much more efficient code. Okay, we are still waiting on the computer to turn on. Yes. Exactly. The 4 families. Yes. Yes, exactly. Exactly. You don't need to wait for. So the output of the second assignment, the correct outputs, the hidden Markov models for these 4 families, I'm going to provide them on May 3rd. Because of the late submission, because it's like the solutions to the second assignment. So I'm going to provide them on May 3rd. But even without them, you can start coding because there are already 2 examples provided for assignment 3. There's the example HMM of this long example, and also this DNA sequence, the short example. You already have two hidden Markov models that you can start working with, on assignment 3 by using these two examples. And testing after you write your program, running it on these 4 families that I'm going to provide is not going to take more than half an hour. All you are going to do is just run your program a number of times, look at the results, and try to put the results on in a table. So in total, there are 10 sequences that in the third assignment. In the third assignment, your goal is to score a given protein sequence against the profile hidden Markov model. In other words, we are going to compute the probability that this profile hidden Markov model generated this sequence. And since we have 4 families, four profile hidden Markov models, 10 sequences, you are going to try each one of these 10 sequences against each of these 4 families. So it's going to be a total of 4 times 10. You are going to run your program 40 times. Here some clue for you is a hint for you. Some of these probabilities are going to be 0. Actually, a lot of them, because you do not use pseudo-counts. Since we do not use pseudo-counts in profile HMM, if, for example, in a column we only see 2 amino acids, and if in our sequence that column has a different amino acid, we immediately going to have a 0 probability of emission from this profile HMM. So if you get 0 probabilities, it doesn't mean that your program is wrong. It means that this is quite possible. And in certain cases, a test sequence may get 0 probability from all these 4 families. In that case, you are going to say that it doesn't belong to any of these 4 families. Okay? I think there's one such sequence which gets 0 probability, or 1 or 2. Out of these 10 sequences, one or 2 of them get 0 probability from all 4 families. And the others, again, another hint for you, it's like they get non-zero probability from the family that they belong to, and they get 0 probability from all the other 3 families. So it's going to be easy to compare prediction is going to be easy. I mean, if you have 4 families and you're testing a sequence against these 4 families, this sequence is going to get a non-zero probability from only one of these. I mean, if my program does not have any bugs, also, I mean, it's quite possible that I have bugs. So we should, after you code your program, you should compare it the results provided in the assignment text. There are 2 probabilities provided in the assignment text, one for the small example, one for the large example. We are going to see if a majority of you agrees with my results. It means that I'm also correct. But since I had so many if-else, it was a really like a complicated code, unnecessarily complicated. It was difficult to debug the program as well, especially for a large input. But I can also check its correctness on the small example, and I'll let you know whether I have any bug in my code. But if you discover any discrepancies with your results, with my results, you can also report them on the news group, and we can discuss. I mean, even in the assignment 2, we had some bugs. The emission probabilities, the first version of the emission probabilities, there was a bug in that. It was counting the gap, but with your help, we were able to correct this. So similar things may happen in assignment 3 as well. Okay, last time we were talking about UPGMA algorithm, phylogenetic tree construction. Where is our, yeah, it's here. This one. And we are going to start protein structures today after talking about UPGMA. We have talked about this algorithm, which if we are sure that the matrix, so as a reminder, the problem what we are dealing with was we have a distance matrix given which shows pairwise relationships between either genes or species, whole genomes. It may be anything. So we have objects which may be some biological entities that may be single genes. We may sometimes try to build phylogenetic trees for a gene, or we may build phylogenetic trees for a whole species. It may be a species tree, gene tree. So your biological object entities may be different things, various things in this matrix. So we have a matrix, a distance matrix or a similarity matrix, which shows the pairwise relationships between these biological entities, between genes. Let's say, let's assume they are genes. So between these genes, by using sequence alignment, for example, we can find pairwise sequence alignment is going to give you the relationship between each pair of genes you have. If you have 5 genes, you are going to have a 5 by 5 matrix which shows the pairwise relationships. And your goal is, in phylogenetic tree construction, our goal is to put these 5 entities in a tree, which shows the hierarchical relationships among them, considering all of these 5 species. Right now we have pairwise relationships. What we want to see is we want to see the global picture. Which of these 5 species are more closely related to each other? Are they, for example, 2 groups that are different from each other within this 5? Also, we want to see phylogenetic tree, we want to see the global relationship between these entities. And if the distance we have here is additive, if it's a matrix distance, meaning that symmetric triangular inequality holds, and we have all positive distances and 0 distance for only identical objects, and then we can use this algorithm which after verification of the distance matrix, we can use this algorithm to build a tree, and the tree we are going to have is going to completely satisfy all the distance matrices here, distance values here. So the tree we have, on the tree, we also have, as again for recap, remember we had 3 distance or path distance. When we put these objects onto the tree, we can talk about the distance of 2 objects on the tree. And our goal is to match the distance on the tree, the path distance, should be as close as possible to this distance matrix, distances given in the distance matrix. So that was the problem we are trying to deal with. We want to construct a tree so that the distances on this tree, the path distance on this tree, is exactly equal to the distances given to us. If the matrix is additive distances, this is possible. We can construct a tree which is going to have the exact same distances as the given distances. But sometimes if our matrix may not be additive, especially if we are using sequence similarity scores are not metric. They do not satisfy triangular inequality, for example. So they are not metric distances. They are not like Euclidean distance. In other words, string edit distance is not a metric. So we cannot, if we have a matrix which is built using the edit distance between strings, we may not always get a tree which is going to give us the exact distances on this matrix. And this problem finding, so if we cannot find the exact tree, our goal would be find a tree which minimize the distances in this matrix and on that tree. So this minimization problem is shown to be NP hard, as difficult as problems that with no efficient known solutions. This means that, I mean, NP hard means that this problem is as difficult as some previously known problems which with no efficient fast solutions. And this means that if you can find an efficient solution to this one, it's proven that it's as hard as these ones. So if you can find a solution to this phylogenetic tree minimization problem, you can also solve the other problems easily. So it's like all these problems are connected to each other in terms of difficulties. So it's called the complexity class. They are all in the same complexity class. They are all equal difficulties. If you can solve one of them efficiently, you can solve all of them efficiently. So they are connected to each other. And this problem is therefore difficult. It's in a difficult problem class. So if we have such a problem, what we do is we use heuristics. Heuristics are just some base, they do not guarantee optimal solution. We do not guarantee to get a tree which is going to minimize the distance between this matrix and the path distances, but it promises to get some intuitive, it's based on intuition. Heuristic algorithms is based on intuition, and they promise good, satisfactory results for certain matrices. And one of these algorithms is UPGMA. I'm going to talk about UPGMA, so I cannot get, okay. Anyway, so UPGMA algorithm is like this. It's the acronym for unweighted pair group method with arithmetic mean. It has a very long name, but the algorithm is very simple. So here, it's an agglomerative clustering technique. There are two types of clustering that you can do. So our goal here, it's a clustering technique means that, so we have biological entities, we have genes, 6, 7 of them, okay? In this example, we have 7 genes. And our goal is to construct a tree among them. And this, how are we going to construct a tree? This tree is going to combine, first, for example, combine, these are the most similar, just combine them first. Then we have some intermediate node here. I'm going to use a different color pen. So I combine them. This red circle indicates their their group. It indicates a group. And then after I combine this, maybe these two are close to each other. I'm going to combine these two. And then maybe these two. Now this group and this may be closer to each other. I'm going to combine these two. And then these two. Okay, so what I'm doing here is that at each step, as you can see, I'm choosing among the available objects, I'm choosing the most similar two of them and combine them into a group. And this is called agglomerative clustering. Starting from the individual objects, try to combine at each step. We try to combine by combining two of them. We are combining groups of individual objects into larger groups. So initially, in the first step, these two were grouped into this group, and then this group was this group included this one. It grew larger. So at each step, for example, maybe in the following step, we are going to combine these two and get even a larger group containing all these leaf objects. So in other words, what agglomerative clustering does is it starts from individual objects, and by grouping them at each step, at the end, it ends up with a single group. At the end, the algorithm starts with 5 or 7, all these individual objects. All these individual objects are in different clusters. They have their own groups initially. Then groups are combined together based on their similarity. And at the end, the algorithm goes until we finally have a single group which combines all the entities. An alternative to this clustering technique would be clustering by splitting the groups. We may assume initially, for example, imagine we have the same 7 entities. And we may think that we may have an algorithm which will assume that initially they are all in a group, in a cluster. Then this algorithm is going to decide, how do I divide this whole group into 2 groups? How do I divide these two groups, this large group into 2 groups? Maybe here like this, okay? Then this is going to give me 2 groups, 2 clusters of 2 nodes and 5 nodes. And then I'm going to maybe divide this one like this one, and this one. So this is also, at the end, this also gives me a tree. but it starts from the reverse side. It starts from the whole entities in a single group and tries to divide each group into two. This is, I cannot recall the exact name for this type of clustering, but it's based on splitting of the one large group into smaller groups. Agglomerative clustering starts from the other side. It starts from each individual member in separate groups and then tries to combine them into larger groups. Here, when we are combining these individual objects, what we have here is we have some intermediate clusters during this process. So when, for example, at this point, after this point, when I decide to put these into a group, I have an existing group in my problem. In this problem, I have a group, and I have another group here. Now, when we are at the next step, imagine we are going to decide which nodes or which groups to combine together. Groups can also be combined together. These are small groups. These red circles indicate smaller groups, which can also be combined to form a much larger group. Okay, so at an intermediate iteration in the algorithm, we need to have a way to compute the distance between two groups. Okay, what is the distance? So the distance between these individual entities, the distance between these genes, are given in the distance matrix. Okay, the distance matrix gives us what is the relationship between these two entities. Now, at an intermediate step, I need to also answer this question. What is the distance between this group and this group? Okay, so this is one important question we need to decide when we are progressing in this algorithm. How can you, given two groups, or imagine at an intermediate point, we have three large groups in our algorithm. Three large groups. Which two of these groups are we going to combine next? Okay, so the algorithm at this step, because the algorithm goes on until we have one final big cluster which contains all the genes. Now, at this intermediate state, I have three large groups. Now, for the next step, I need to decide which two groups to combine next. So, in order to be able to do it, what do I need? I need to have the distances between these three entities, right? I need to have, what is the distance between this group and this group? What is the distance between this group and this group and this group and this group? I need to compare these three distances and pick the minimum of these three distances. If the minimum is this, I'm going to choose to combine these two to a larger group. So this algorithm needs a distance between clusters, and in UPGMA, this cluster distance is defined very simply. It's the average of all the distances between individual cluster members. It's like if this cluster contains 3 members, if this cluster contains 2 members, the distance between these two clusters are defined as the average of these distances, average of these 6 distances. Okay, if this one has 2 elements, if this one has 3 elements, I can have 3 times 2 different pairs of mappings between this. I mean, I'm going to look at the distance of, so if this is A, B, this is 1, 2, 3. I'm going to look at the distance of A1, distance between A2, distance between A3, distance between B1, B2, B3, and take the average of those. Just add them up and divide it by 6. The average of these distances is the distance between these two clusters. And formally, it's this. You sum up all the pairwise distances between an element that belongs to cluster A and an element that belongs to cluster B. And the total number of such distances is going to be the number of elements in A times number of elements in B. So you divide it by that to get an average of this distance. So here's an example. We start with 5. So there are 5 genes, okay, 5 sequences that we are going to build a phylogenetic tree. We are given this distance matrix. And the algorithm proceeds like this. At each step, we are going to combine the shortest distance, the pair of objects which has the minimum distance. If there are ties, meaning that if there are two pairs of objects with the same minimum distance, we break these ties randomly. I mean, the algorithm does not regularize this. I mean, it just picks one of these pairs randomly if we have equal distances. As in this case, these two distances between BC and BD, they're all 4. We can either choose BC or BD at the first iteration of the algorithm. They're both equal. I mean, the algorithm may choose either one of them randomly. So what we do is, we first try to combine BC or BD. Let's see what we are going to do. So the algorithm, this particular run chose to combine BC. After combining BC, what we have here is we introduce a new node here, a group node, which represents the group which contains B and C. Okay? And we update our distance matrix. We remove B and C from the distance matrix, and we put this group instead. Okay? At each iteration, our matrix is going to decrease its size by 1. If it was initially a 5 by 5 matrix, now it's a 4 by 4 matrix because we chose to combine B and C. And we remove B and C from the matrix. We inserted the group BC here. Now, after inserting this group BC, how are we going to update the values in this matrix? So what is the distance between A and BC? Okay, so we need to decide that by, for example, here, the distance between, so we have two groups. One of them is just A, and we have another group which contains B and C. Now we need to decide what is the distance of A to BC. And the algorithm says you just take the average, DAB plus DAC divided by 1 times 2 divided by 2. It is going to be 11. 10 plus 12 divided by 2, it's going to be 11. So we do this for all the entities in this matrix, and we get ourselves an updated distance matrix like this by just taking the average. And another thing we need to decide is, after updating the distance matrix, so we chose to combine this, another thing we need to decide is what is the path length, what is the edge weight of these branches. So we combined it, made it an intermediate node BC, so we are building a tree. What is the branch length from BC to B and BC to C? So we need to also decide on that. And the UPGMA algorithm just divides the distance between B and C equally among them. So the distance between them was 4. So this 4 is divided into 2, and these branches are distributed to these branches as lengths 2 and 2. Okay? It preserves the length between B and C. And we never update these values. After we decide that these branches are going to have lengths 2 and 2, they stay like that. Okay? The algorithm, the tree is fixed, that part of the tree is fixed from now on. Now, let's look at the second iteration. What we see is when we look at the distance matrix, we see the 5 is the minimum distance. So what we need to do is combine BC with D. So we combine BC with, now here, combine BC and D. Okay. After combining BC and D, what was the distance between BC and D? It was 5. So it's divided equally, like 2.5 and 2.5 among all the members. But this 2.5, that distance 2.5, is computed as the distance that goes all the way to the leaves. Okay, so in other words, so this leaf node here is D, so it gets 2.5. However, the distance to the leaves is, we already have 2 here, so the total path length to a leaf node should be 2.5, so we give 0.5 between BCD and BC. Okay, so we divided 5 equally and we distributed the edge weights like this. And we update the distance matrix again. We remove BC from the matrix, we remove D from the matrix, and we put BCD to the matrix. And we again use just arithmetic average to update these distances. The next is A and E, which with distance of 7. And it's simply like this. We combine A and E, 3.5, 3.5, and then our matrix becomes like this. We only have 2 groups left, as you can see, AE and BCD. The only thing that is left is going to be combine these 2 groups in the final step, AE and BCD. The distance of 12 is, so 12 is the 6 and 6. The path from this root node to the leaf node is going to be 6, and from this root node to all the leaf nodes is also going to be 6. And this is the final produced tree by UPGMA. So this tree is called an ultrametric tree because every leaf node in this tree is of equal distance to the root node. All of them has equal. Since we had this divide by 2 and we maintain this distance to the, by adding new things, we were just adding equal amount of things to each path length. So if you look at here, every leaf node here is of distance 6 to the root node. This is a very special tree. And this specialty of the tree actually makes it, it cannot represent all kinds of pairwise relationships given in the original distance matrix. Such a distance is a very special form of a tree, and the distances between objects are ultrametric distance as well. This produced tree implies ultrametric distances between these objects. For example, if these 3 objects, which are on different parts of this root node, these 3 are on these branches and these 2 are on these branches, they all have equal distances between them because their distance to the root is the same. The root from that leaf node is the same. So this BCD is going to, all of them are going to have an equal distance to A and E, which is not 6, sorry, 12. Which is going to be 6 times 6 plus 6, 12. The path distance is going to be same for all of them. But what you notice here is that the path distances here is not the same as the distances that we were given in the first place. It shows that this UPGMA algorithm does not guarantee you to find a tree which satisfies the original distance matrix. They are close. You are going to get a close tree, but they may not be exactly equal. Let's check, for example. The distance between B and C is 4. Yeah, it's true here. The distance between B and D was 4 originally, but here the distance between B and D is 5. Okay, so we have a certain error here. In the original distance matrix, it was 4, but in the tree here, it's seen as 5. And here, the distance between E and B was 14 in the original distance matrix, but in our tree, it is 12. Okay, so there are certain discrepancies, and we can actually compute how close we get to this original distance matrix by producing another matrix, which is called the tree distances. Let me just have this 5 by 5 matrix. So A, B, C, D, E, A, B, C, D, E. This is going to be symmetric, so I'm just going to fill in the upper triangle. And okay, let's do it. So these are all 0. The distance between A and B is 12. A and C is 12. A and D is 12. A and E is 7. The distance between B and C is 4. B and D is 5. B and E is 12. The distance between C and D is 5. C and E is 12. D and E is 12. This is the matrix we have produced by building this tree. If you compare it with, I mean, it's a very regular matrix, as you can see. There are the two main groups, AE and BCD. The distance between all the individual members are all 12. Okay, and even in the subgroups, the distance between BD and CD, it's all 5. They are also equal. Okay, so it's this is an ultrametric matrix, and if we look at it, we see we did not actually do a bad job in approximating the matrix. If you look at the results, so we have 12, 12, 12, 7, but these were also high numbers, okay? 10, 12, 8, 7. I think the worst is between A and D. It was 8, we increased it to 12. This was 4, 4, 14, we made it 4, 5, 12. 5, 6, 16 is 5, 12, and this 12 is correctly found. So it approximates the original tree. So it's, as you can see, how a heuristic algorithm provides an approximate solution. Okay, the actual, if this was not an additive, actually this is an additive tree, and there exists this is the actual tree. As you can see, this was the produced tree. If you look at the actual tree, you see that in the actual tree, these branch distribution, the distribution of these weights to these branches should not be made equal. I mean, we just distribute equally 2, 2, but in the actual tree, B is actually closer to other objects compared to C. C is more farther away. If you compare B and C, whether how close they are to the rest of the objects, B is more closer to the rest of the objects compared to C. So when you're distributing these weights, you can take into account these, what is the overall distance of B to the rest of the objects, and what is the distance of C to the rest of the objects, and divide the weight accordingly. This is what neighbor joining algorithm does, but I'm not going to talk about the details of neighbor joining algorithm. So an actual tree, I mean, it's possible to have a tree with the same structure, same topology, but just the weights on these edges are different. By just updating the weights on these edges, it's possible to get a tree which is exactly the given input matrix. And neighbor joining algorithm guarantees to find that tree if your distance matrix is additive. If there exists a tree, neighbor joining guarantees that. But if it's not, if your distance matrix in the original distance matrix given is not additive, I mean, if such a tree never exists, both of these algorithms promise to, I mean, they produce a tree which approximates the results. You may not get an exact tree, but it's going to give you a tree which looks similar like the distance matrix given above. They are very efficient, very fast algorithms, as you can see, with linear on the time of, actually at each time we are doing n-square comparisons. We are computing distances between all pairs of objects. If you look at the overall complexity, I think it's going to be somewhere along n-square or n-cube. I don't think it's going to be n-cube. We need to do a detailed analysis of that to find the actual complexity. These are the limitations of UPGMA. We are going to get the exact tree if the distances given in the first place were ultrametric. Okay, I'm going to skip neighbor joining. I'm not going to talk about neighbor joining algorithm. And there are lots of existing tools that you can use to build these trees, phylogenetic trees. Philip is a popular application. There are others which you can do. You can also, the neighbor joining plot, it gives you a visual representation of the produced tree. Now I'm going to do an introduction to protein structures in the remaining 5 minutes, and I'm going to we are going to continue protein structures tomorrow. So now, up to now, we all talked about pairwise or distances between sequences. So sequence data was the main data that we have talked up to now. Even these in the phylogenetic trees, the similarity distance matrix, we computed using pairwise sequence alignment. Now we are going to talk about different types of data, a different type of data, which is going to bring in different types of problems. So a protein structure, this linear sequence of amino acids in the cell is actually, the protein is folded into a three-dimensional shape in the cell, in the 3D space. It has a three-dimensional shape composed of atoms. And these atoms, they are going to have form a molecular surface. So when we talk about protein structures, we are going to be talking about strange-looking shapes like rocks, okay? Or the thing is, I'm not going to be able to show you a real picture of a protein, because it's not possible to just take your camera and even with the electron microscope we cannot see them with real light, because they're atomic level structures. So what we are going to have is some visual descriptions, like cartoon-like descriptions, or there are several ways to show the shape of a protein. And each of them shows different aspects of this structure. You can, for example, by using spheres, you can represent each atom by the van der Waals volume as a sphere, and you can put all these spheres in 3D space and visualize your protein as a set of balls, as a set of spheres. Okay, that's one representation. Or you can approximate how this surface is going to interact with other atoms. You can also provide a molecular surface, which is more like a smooth surface. You can have a mesh structure, a triangular mesh, which shows the molecular surface. Or you can just visualize the backbone of a protein structure. I'm going to talk about what a backbone is by just considering the peptide bonds, the nitrogen, alpha carbon, and carbon atoms, the connection between these atoms, which provides the linear connection between linear chain of amino acids. You can visualize that backbone, which is going to give you another idea about the protein structure. So there are different ways of visualizing protein structures, but in essence, at all of them, they are three-dimensional data. We will be now working on data which is the three-dimensional shape, like this eraser, like this pen. Okay, so we can identify this eraser by sampling points on it and by providing the XYZ coordinates of these points on the Cartesian coordinate space. Okay, so how do you represent this? We are going to be talking about how do you represent this eraser? Okay, you want to digitize this. This eraser, this three-dimensional object, how are you going to represent it on computer? What can you do? If you just take a photo of this eraser, that's two-dimensional data. How are you going to get the depth of this eraser from that two-dimensional photo? So what we need is a three-dimensional digitized version of this eraser. How can we do that? As I said, one way is we first choose ourselves a reference coordinate reference frame, which is we need to decide if I'm going to provide some XYZ coordinates for points on these eraser. I need to define where is the origin, where is the x-axis, where is the y-axis, where is the z-axis. I need to define myself a reference frame. For example, this corner here becomes my origin 0, 0, 0. This is my y-axis. This is my z-axis, and this is my x-axis. If I define orthogonal axes, then I can this point here, for example, is going to have a certain XYZ coordinate. I can define uniquely the location of a point on this eraser by a triplet XYZ. And if you can sample enough points on this eraser, and you just list those coordinates of these sample points. You can compute the volume of this eraser, the depth of this eraser, the shape of this eraser, the corners, the angles, everything can be computed by these XYZ, the set of XYZ triplets that are sampled from this eraser. So point cloud, sampling points on this object is one thing. But we can do other things as well. We can maybe represent this object as a set of some polygons. Instead of points, I can divide this eraser into smaller polygons. Or, for example, there's a surface, there's a plane, this ellipse-like looking surface here. I can have this equation of this ellipse. I can also have equation of these parts. I can also represent it as a set of polygons. That's another approach. So for proteins, what we are going to do is we are going to represent each protein, the popularly accepted format for protein structure is that we are going to have the coordinates, XYZ coordinates, of each atom that this protein contains. So at the atomic level, we are going to say that this carbon atom is at this XYZ. This nitrogen atom is at this XYZ. So we are going to have, if you go to protein data bank and download it's freely available, by the way. You can go to protein data bank and download these XYZ coordinates for your favorite protein. If your favorite protein is not among the structurally among the 50,000 proteins whose structure are known, you're going to be sorry about that. I mean, out of millions of proteins, only we know these XYZ coordinates for only 50,000 or 50 or 60,000 of these proteins, but 60,000 is still a large number. You have like shapes that you can play with, 60,000 such shapes. You can do a lot of analysis on them. So we are going to be talking about these, and the main problems in protein structures we are going to be talking about is, given two protein structures, how can you compare them? So this the same analog problem in sequence alignment. Given two sequences, we were wondering how these sequences are similar, how they're related. Now the question becomes, given two shapes, if I give you a set of XYZ coordinates and another set of XYZ coordinates, can you tell me whether these two protein structural shapes are similar? For example, one protein may be a long helices. They are going to be elongated protein like this. Another is like structural ball-shaped protein. So by looking at these, you can say, oh, okay, these structures are not similar. So by manually visualizing these structures, you can say, okay, they are similar or not. But the problem is, how can you do it automatically? How can you write a program by taking these two sets of XYZ points to determine whether these two shapes are similar or not? If they are similar, which parts are similar? Maybe overall they are not similar, but there are certain substructures which are similar between them. So this is the problem of structural alignment. We are going to talk about that, and I'm going to talk about for a week how do we determine these structures computationally. There are experimental, the structures in the protein data bank are determined experimentally, but there are also tools that, I mean, that's the first problem that comes into one's mind is, we have 7 million protein sequences. Didn't anybody try to predict, I mean, given a sequence, why can't we find its structure? Okay, do we have tools for that? And that's one of the biggest still unsolved problems of the century, in the protein folding problem. And I'm going to also talk about that problem. Given a protein sequence, ordered list of amino acids, how can we predict the structure, the three-dimensional shape this protein is going to have? So the main two problems we are going to talk about in protein structures are going to be the structural alignment and structure prediction. Okay, so let's see you tomorrow. If you have any questions about the assignment, the new assignment, you can email me or post it on the news group.